All right, hi everyone. Welcome back to the third and final session on single cell RNA-seq data analysis. So, as always, let's begin by navigating to our project directory associated with these meetups. And if you don't have one, just open up our studio. And in the console, once again, we type in remotes install underscore GitHub Duke CCT slash CBRG. And we specify that force is equal to true. And we do this so that we install the package that we use for these meetups, or reinstall the package if you already have this package, if you've previously installed this package. Now, this is meetup 15, so over the last many weeks, this package has grown in size. So as you can see, it'll take a little while to download, hopefully not too long. While it's downloading, I will briefly describe the objectives for today. So as I mentioned, this is the final in our three-part series on single-cell RNA-seq analysis. And last time we went over computing various quality control metrics. And the purpose of this was to filter out low quality genes and low quality cells. It was a longer than usual session, and much of the work that we did was admittedly a little tedious and boring, but it is important. Fortunately for today, we'll be using those pre-computed quality control metrics. So this week we'll be doing the fun stuff of deriving some kind of biological insight from our single-cell RNA-seq data. All right, so this is installed. Hopefully it's installed for you also. And after it installs, just load the package as you would with any other package, library CBRG, and then launch today's our markdown file. So it's meetup 15, so we do launch project 15. And once again, as always, we begin by running the setup chunk. And so this will look to see if you have these packages installed. If you don't, it will install these packages, and after installing all the relevant packages, it will load all of them. Once again, all of this very nice code was written by my colleague Paul. I should say, again, if you want to annotate this our markdown document, which you should, which I encourage you to, recall that you have to save as, unfortunately. So if you just save this R markdown file, it'll save it in a directory that contains this package, which is kind of hard to get. So if you want to save it within your project directory, you have to do save as, just as I did. And so I can see that project 15, the one for today, is now in my meetups directory. All right. So just below the setup chunk, there are some links to hopefully useful resources. So the first link is to the book that we've been using. So once again, this is a book that was that is associated with a single-cell RNA-seq course that was run by the Sanger Institute a few years ago. And we will specifically be going through sections in chapters 6 and 7. If you would like to download the files separately for all of these analyses, you can do so from here. We don't have to, but if you would like to have these files on your machine, this is the link to go get them. And finally, these data correspond to a published study by Tung et al. And the purpose of this study was to look for things like technical artifacts and batch effects. And those are the things that we will actually be focusing on today. And this was published in, I believe it was Scientific Reports. And this is a link to that paper that describes the study in detail. So the first thing we're going to do is we're going to do a normalization step. And we're going to use this approach called scran. Before we go into why we need to do normalization and what scran is, let's first reload our data. So if we run line 96, this will import the data set that we prepared in the previous session, and we'll save it as an object called UMI. Just to quickly remind you of what UMI is. So UMI is what's known as a single cell experiment object. So let me first clear the console. If you want to take a look at UMI, one thing you can do is just simply type in UMI and take a look at it. And you get a little bit of information about what this object contains. So it contains 18,092 rows, so these are the genes, correspond to genes, and 864 columns, so these correspond to individual cells. You can see that this single cell experiment object contains two assays. Counts, so these are the raw counts, and log counts raw, so these are the log transformed counts. And this is something that we did in the previous session. We log transformed these data. And today we'll see why a log transformation might be useful. And then there's some other information here. This is fine, but it's limited. Recall that for these kinds of objects, so like for the kinds of objects we looked at when we were doing a proteomics analysis or RNA-seq analysis, these objects often have data or metadata associated with the rows and the columns. So recall that the rows correspond to the genes. So if we wanted to learn something about them, we could just type in row data and then the name of the object. And if we run this, we see in our console that there's quite a bit of information. We see, so once again, the columns correspond to genes. So these are the ensemble IDs of the genes. And then the columns have some information about these genes. Importantly, the symbol, the mean count across the cells, the percentage of cells in which these genes were detected. And if you're curious to know how these columns were computed, this is what we did. This was some of the work we did in the previous session. Importantly, rather the most important column in this data frame is this column here, discard. And discard is just a Boolean column, so it's composed of either falses or trues. If it's a false, that means we keep that gene. And if it's a true, there was something wrong with that gene. So again, as we discussed in the previous session, there are many things that could be wrong with a particular gene. One thing is maybe it's not expressed in that many cells. So it's not really going to contribute much to our analysis. So we can just get rid of it. And in fact, you can see, look at these two trues, the genes that have trues in the discard column, we see that the mean expression is tiny. It's effectively 0. So we will be using this discard column this information in the discard column to discard these suspect genes. Similarly, if we wanted to find out something about our cells, we can do call data, UMI. We run this, again, we get another data frame. It's pretty big. Actually, it's really big. But unlike the row data frame, here, in this case, the rows correspond to the individual cells. And we see lots of other useful information. We see which individual. So there are 9 individuals in this study. So we can see which individual a given cell belongs. There are replicates. So I'm sorry, there are not 9. There are 3 individuals. My mistake. And each individual, for each individual, there were 3 replicates. So this is where this study design allows you to look for things like technical artifacts and batch effects. And there's lots of other information that, once again, we computed in the previous session. We can, for today, ignore all the rest of the information except for this final column here, which is, again, discard. It's slightly confusing because there's a discard column for both the genes and the cells. But the meaning is the same. Once again, if discard contains a false, that means we want to keep that cell. It means that cell is fine. But if it contains a true, for whatever reason, we want to get rid of that cell. And as we discussed in the previous session, an example of one of these reasons could be a higher than average or an unusually high amount of mitochondrial DNA. And this is something that is cause for concern. So these cells are good to get rid of. Once again, we define the thresholds and things like that, and those details in the previous meetup. For today, we will just be kind of blindly using this discard column information to subset our UMI single cell experiment object. So that's really a quick fire recap of what we did in the previous session. So how do we actually subset? So this is something we have taken a look at a couple of times, but to remind you, So if we go to line 104, this is where we're supposed to enter our code to clean up our UMI object. And what that means is we get rid of the genes that we're supposed to discard, and we get rid of the cells we're supposed to discard. So recall, to subset a single cell experiment object, we can use the square brackets notation, just like with a data frame. And within that square brackets, we put in a comma. So anything to the left of it corresponds to rows. Anything to the right of the comma corresponds to columns. So so I mentioned that we can use this discard column for both the genes and the cells. How do we access it? So if you look at 100, line 100, we can use row data on the UMI object to extract the gene information corresponding to the gene. But we don't want all the information. We just want what's in the discard column. So to do that, that's easy, which is we just do $discard. And to confirm that this in fact just extracts the discard column, we can copy paste row data UMI $discard into the console, and we just get a bunch of trues and falses corresponding to the discard column. And we can do the same thing for the columns. The only difference is that we do call data UMI, and once again $discard. And recall that we've used this approach previously where we've used these trues and falses to subset data frame, vector, list, whatever. Now, if we just subset this UMI object like this, we will end up retaining all the genes and cells that we're actually supposed to discard. So to do the opposite of that, to actually keep the quote-unquote high-quality genes and cells, we negate both of these things. And to negate something in R, you can use the exclamation mark. So we negate both of those things. Now, we need to assign this resulting object to another object, which we can call umi.qc. That's the term that the book uses. So if we run this, we now see that we have a new object called .qc. You can take a look at it. Not sure if you recall the dimension. Well, actually, let's look at UMI itself and quickly take a look at its dimensions. 18,000 genes and 864 cells. But if we look at UMI.qc, we see that it has 13,800 genes and 670 cells. So we can see that we've gotten rid of quite a few genes and cells. So that's it. That's the quality control step. Super easy today because we spent 90 minutes computing these various metrics in the previous meetup. But once you've computed them, cleaning up your single cell experiment object is super easy. Okay, so now we start to come to the point where we can consider things like normalization. Why would we even consider that? Well, I mentioned that this dataset contains information for 3 biological samples, and each of those biological samples has 3 technical replicates. So what we would expect is if we use some kind of visualization approach like principal component analysis, we would expect or hope that the cells that correspond to the biological replicates all kind of cluster with each other. And there's a separation, hopefully, between these 3 biological samples. What we don't want to see is separation within the biological samples of the 3 technical replicates. If there is any separation, this is an indication of some kind of technical artifact or batch effect. So how do we visualize this? I just mentioned PCA, so we'll use PCA. So running PCA on a single cell experiment object is super easy. What you do is you use the dedicated function run PCA, and then you give it, the first thing you do is you give it the name of the single cell experiment object. And then there's this additional parameter called xprs_values. And this is what you use to specify which assay you are performing PCA on. Recall that the UMI.qc object contains 2 assays, counts and log counts raw. So this prompt on line 107 is asking us to perform PCA on the counts data. So we just do exprs underscore values is equal to counts. And we need to reassign this to the umi.qc object. Before I run this, if you look at the console when we ran umi.qc, there's one slot called reduced dim names, and it has a value of 0. So this is where, after you perform PCA, this is where the resulting principal component information is stored. So right now, UMI-QC does not have any such information, so there's a 0 there. But if we run this, run PCA, and take a look at UMI.QC again, we now see that there's a reduced dim names 1, and it's PCA. So it's even telling us what kind of dimensionality reduction we performed. We can do other kinds, like we can do t-SNE, for example. We'll stick to PCA for today. So once you've computed, once you run PCA, you can now plot the principal components. So that's, again, super easy. There's a function called plot PCA. Once again, it takes in this single cell experiment object, umi.qc. And actually, we can just run this like this. And we get this plot, which is fine. It gives us useful information. So principal component 1 accounts for 78% of the variation. Principal component 2 accounts for 9%. That's fine, but it just looks like we have a bunch of gray circles. So fortunately, there are multiple parameters that we can supply values to for this function to color these data points in some kind of useful way. So one parameter, if we go back to line 109, is we can use the color by parameter. So if we go back to, we do call data umi.qc. So we can, the values that we supply to these parameters for the plot PCA function, we derive those values from this call data output. So we can, for example, color, I mentioned there are 3 replicates. So we can color these data points based on which replicate they correspond to. So we could do color by is equal to replicate. I just noticed that the spelling of color here is with a U. And I think this package was developed by people at Cambridge. So apologies to all the American viewers. And so just as we can specify the color, we can also specify the size of our data points, and we'll see why this is useful to do. So we are going to specify the size based on the number of detected counts. So this is also known as the library size. And we'll see why this is a very useful thing to look for and actually to do routinely when you are analyzing single-cell RNA-seq data. And the final parameter we will set is the shape by parameter. And we can shape it by individual. So I mentioned that there were 3 individuals in this data set. So if we run all of this, we get this much more informative PCA plot. And if we take a closer look at it, we can see that the replicates, R1, R2, and R3, they're colored. They have 3 different colors: blue, orange, and green. And then we set the size of each of these points to correspond to the number of detected counts. So we can see the smaller the size, the fewer the counts, and vice versa. And finally, we set the shape of these data points. So circle, triangle, or square to correspond to the 3 different individuals. Now, it should be clear, it is clear, when we look at this PCA plot using just the raw count data, there's really no separation, which is bad. We expect, as I said, the points corresponding to these 3 individuals who are 3 separate biological samples to cluster with each other and be separated from the other 2. We don't see that. We just see this huge clump. Another thing that isn't particularly clear from this plot is, if you look carefully, you'll see bigger shapes to the left and smaller to the right. So this is actually, unfortunately, a very common occurrence in single-cell RNA-seq data. Having the first principal component, so the most important one, being largely driven by library size. And that's what you see here. You see this nice separation of high count cells from low count cells. And this, you don't want to see that, because this is a classic example of a technical artifact. Okay, so clearly our unnormalized raw count data are kind of useless at telling apart biological replicates or biological samples from each other. What does log transformation do? If we look at line 112, we see that this is very similar to the previous code we ran. The only difference is we set the exprs underscore values to equal log counts raw and not counts. And then we reassign this back to umi.qc. One thing worth mentioning here, so if we run umi.qc in the console, and once again take a look at the reduced dim names slot, again, we just see the number 1. Now, this is at first slightly unusual behavior, because while this object can hold more than one assay, so right now it's holding 2 assays, it can only hold one product of dimensionality reduction at a time. So if you run line 112, as we just did, the previous principal components corresponding to the raw count data, they get replaced. So that's just something worth bearing in mind. Now, if we run line 113, we get a plot for our log transform data. A few observations. One, if we look at the principal component, the first principal component, it's gone down from accounting for 78% of the variance to just 19%. And recall that the first principal component was largely being driven by library size. So this is kind of like a correction already. It's not a perfect correction because once again, you see separation based on size. You see the big rectangles and squares here and the small rectangles and squares and circles here. And that's something we need to correct. One thing that is good to see is you start to see some separation between the biological samples. So from here, we can see that the individual who corresponds to the square symbols, they are here. The individual that corresponds to the triangles, they are here. Unfortunately, there's an overlap between the circles and the triangles, so there's not a good separation of those 2 biological samples. But still, this is a step in the right direction. So that is just an introduction to how we can use principal components to visualize our data and the effects of any kind of transformation. So in this case, log transformation. So can we improve this? Yes. So one way to do that is to perform some kind of normalization. So we actually encountered this when we were doing RNA-seq, bulk RNA-seq analysis. And recall that for various technical reasons, you can have some samples that just have greater library sizes. So just in general, across the genome, you have more counts for each gene. So now, if you were to compare these samples with other samples that have smaller library sizes, you can get spurious results, like spurious differential expression analysis results. So what we did was we accounted for these differences in library size by performing normalization using the DE-seq2 approach. So we could just use that approach, we could apply that approach to the single cell RNA-seq data, because now instead of having samples, we have individual cells. The cells are our samples. The reason this fails, as I've described, as I've noted here on line 121, is because single cell RNA-seq data are composed of a huge number of zeros. And one of the drawbacks of the DE-seq approach is, for one of the steps, we have to calculate the geometric mean. And if you have even a single zero, the resulting geometric mean will also be a 0. So if we were to use a DE-seq normalization approach, we would have to ignore all the cells that have zero counts for a given gene. So we'd be discarding a huge amount, if not most of our data. So to address this problem, there was this approach called scran that was developed specifically for normalization of single-cell RNA-seq data. And here's a link to the paper that describes this approach. There are quite a few details, and as you can imagine, the paper is quite heavy, maths heavy, but it's really well described, even for a paper that uses a lot of maths. So just briefly, the approach is described here on line 123 onwards. So the first thing we do is we cluster cells based on library size. And there are various technical reasons for this that are described in detail in this paper. Within each cluster, so step two, the algorithm, it kind of divides these clusters into smaller pools of cells. And these pools are overlapping. And then what it does is it sums up the gene counts for each of these pools. It then normalizes those pools to a reference so-called pseudocell. And that reference pseudocell is simply an average across all gene expression for all the cells. So in this way, it computes what it calls a pool size factor. So each of these cell pools now has a size factor that gives you information that is related to the library size. Using this pool size factor information, they perform this thing called deconvolution. And again, the details of this are in the paper. But basically, it's just a huge system. By creating all of these pools, what you end up doing is you end up creating a huge system of linear equations. And so what the last step, the deconvolution step, does is it solves those linear equations to estimate size factors for each cell. Because recall that a pool size factor is composed, can be thought of as being composed of multiple individual constituent cell size factors. Now, please pardon the pun, this seems like a somewhat convoluted approach, but in the paper, they clearly describe why and demonstrate how this approach is more robust than other normalization approaches. So anyway, those are all the gory details, and which I welcome you to go through in this really nicely written paper. Fortunately, actually implementing this is super easy. So if we go to line 130, recall that the first step of the scran approach is to compute these clusters, which is easy. We use a function called quick cluster. We give it the name of the single cell experiment object, and then we have to specify the size of this cluster. Why is it min size equals 30? Honestly, because that is the hyperparameter that was chosen in the book. So I'm just sticking to 30. I think, unfortunately, these are one of those things that you have to test. You test a range and see what gives you the quote best result. So let's stick to 30. And we assign this output to an object called qclust. We then need to, using this information, recall that we have to now compute these size factors. First for the pools of cells, and then for the individual cells. All of this is done using this function called compute sum factors. And that just requires the single cell experiment object and this clusters information. So clusters is equal to qclust. And then we can assign all of this back to the UMI.QC object. And if we run this, this will take a few seconds to run. Well, actually, just a couple of seconds. And now, if we go to line 132, if we look at the column data for UMI.QC, once again, we have lots of information for each of our cells. But if we look at the last column, we see we now have a size factor column. So we now, importantly, have computed a size factor for each of these cells. And we can use this size factor information to normalize our count data. And this normalization takes into account or corrects for differences in library size. And all of that is done on line 133 using the log norm counts function. And now, so we apply that to UMI.QC, assign it back to UMI.QC. Now, if we look at UMI.QC, notice that we have a third assay. So we previously had counts and log counts underscore raw. Now we have an assay called log counts. And this log counts contains this scran normalized count data. So has this done anything? Let's run PCA to find out. So if we go to line 138, once again, we run PCA, but this time noting that we are performing PCA on this new log counts assay. And if we plot that PCA object, sorry, if we plot that PCA plot, we get this. So this is the PCA plot for our just the log transform data. This is the log scran normalized data. This looks a lot better than this. For one thing, notice that we go from PC1 accounting for 19% of the variation to just accounting for 9%. And we no longer see this small to big kind of continuum. Also importantly, here we have a pretty good separation of two biological samples, but not the third. For the scran normalized data, we have nice separation of the three biological samples, so triangle, circle, and square, into three different kind of clusters, which is great. Again, this is not perfect because we see within our square individual separation of these three clusters of cells. So these are the three technical replicates. Again, we shouldn't see separation because those are, after all, technical replicates. So any separation between these three, we can assume is being driven by technical reasons. So, and we will apply another strategy to see if we can continue to get improvements on this already very nice plot. So this is using principle PCA to assess the effects of whatever normalization approach we're using. There's another approach, and it's called this Rle plot. So Rle stands for relative log expression plot. So I will make these plots and then briefly describe what these plots are. So to plot an Rle plot, it's really easy. If we go to line 143, we see again there's a dedicated function for this. It's called plot Rle. We put in the name of our single-cell experiment object, and then we give the name of the assay that we want to use to make this Rle plot, and then we can color by whatever. I'm going to color by replicate here. So if we run line 143, so the way to interpret an Rle plot is, what the Rle plot is doing is it is plotting transformed data in such a way that each point here, so what this is this is actually, it's hard to tell, but this is a collection of box plots. It suggests that we have so many samples, so many cells, so each box plot is just basically looks like a line. The thing with an Rle plot is, if there is no, and it's based on the big assumption that most of the genes between two samples or cells, most of the genes show approximately the same level of expression. So this is a big assumption of these Rle plots. If that assumption holds, what you expect to see is, if you compare one box plot with another, you should see a very similar mean around 0 and a very similar spread. Now, looking at this plot, we can see that there are a few samples that violate this. Many samples do in fact have a mean of 0, but many others have higher means. So this is one way to see if there's something fishy with your data. Now, if we plot an Rle plot for the log transform data, we actually see it, actually in some respects, looks worse than just the raw count data. However, if we look at our scran normalized data, this is what an ideal Rle plot looks like. So this is an alternative way of visualizing, identifying technical artifacts in high-dimensional data. So just briefly, a brief description of what relative log expression plots are. Here's a link to the paper that describes these Rle plots, and once again, really nicely written paper. And Rle plots were initially developed to assess the role of technical artifacts in microarray data, but can be applied to any high-dimensional data. So, as they say here, unwanted variation can be highly problematic, and so its detection is often crucial. Rle plots are a powerful tool for visualizing such variation in high-dimensional data. Rle plots are particularly useful for assessing whether a procedure aimed at removing unwanted variation, that is, a normalization procedure, has been useful. And that's exactly what we use this for. So this is the log transform data, pre-normalization, and this is post-normalization. So our Rle plot obviously tells us that normalization had a beneficial effect. And they define unwanted variation as variation caused by technical factors and not by the biology of interest. Briefly, the procedure involves, for a given gene, calculate the median expression across all samples, then compute the differences between expression in each sample and this median, and you repeat this for all the genes. And then for each sample, you make a box plot, as you can see here, multiple box plots, for all of these deviations, for each sample. And again, the big assumption here is that expression levels of a majority of genes are unaffected by biological factors of interest. Once again, I strongly encourage you to read this paper where all of what I said is described in a much better way. But the take-home message is, this is bad, this is good. So the final kind of correction that we'll be performing is batch correction. So for batch correction, if you have any information, any kind of batch information, for example, let's say you are pooling information for a particular experiment from for a particular project from experiments that were conducted in multiple labs. So in this case, the batch would be the different labs. Or you were doing an assay over a few days. So date would be a batch. So if you had some kind of batch information, you can use this approach called combat that briefly it takes into account this batch information as an additional covariate in a regression model. So it accounts for the effect of this batch when it's computing whatever it is it's computing. The details are a little bit fancier than this, and they're described in this paper. In this particular case, we will use replicate as our batch covariate, and that's because that's the replicate that was used. Sorry, that was the batch covariate that was used in the book. I'm not entirely convinced, given the description of this experiment in the paper, that this is strictly a batch. But rather than go into why I think that, I'm just going to mention that as a potential caveat. But we'll just go ahead while acknowledging that caveat and just run combat anyway, using replicate as a covariate, just to see how it works. And so to run combat, you just use this combat function, capital C, capital B, and then we give it the name of we give it the assay we want to do batch correction on. And that is the log counts information. So this is the scran normalized information. One way of extracting that assay from the single cell experiment object is to use this dedicated function called log counts. Because if we go back to just type in umi.qc in the console, we see that the assay that contains the scran normalized data is called log counts. So there's a convenient function called log counts that can directly just get that assay out of umi.qc. And to show you what that means, if you just copy paste log counts umi.qc into the console, you get, UMI. Let's take a second to run. We get the scran normalized count data for the genes and cells, UMI. So that's the assay we want to perform batch correction on. And then I mentioned that we need to specify our batch covariate. That's easy enough. We can just do umi.qc, and then we can just extract the relevant column. So that's replicate, just using this dollar symbol. We need to assign, we can assign this back to umi.qc slightly confusingly. So previously when we assigned the output of our principal component analysis, we just assigned it directly to UMI.QC. We can't do that with Combat. What we have to do is we have to assign it specifically to the assay slot. So we do that by doing assay UMI.QC, and then we need to give a name to this assay, which we'll just call Combat. We run all of this. So it'll take a few seconds to run. Right, so it's performing this batch correction. It's found 3 batches because there are 3 replicates, and it's done it. So now we can look at the effects of this batch correction using Combat. So to assess this effect, let's first remind ourselves of what the PCA plot for just the scran normalized data look like. So pretty good plot. This is what we saw before. Now let's look at the effects of batch correction. This is slightly better. See, so here with the non-batch corrected data, we have nice separation of our biological samples, but a bit of separation of our technical replicates for this biological sample. After performing batch correction, there's still a little bit of separation, but it's smaller. And I can somewhat confidently say that because if you look at the percentage of variance accounted for by the first principal component, that stays the same, 9%. So this, yeah, it visually looks like it's made a mild improvement. Similarly, instead of doing a PCA plot, we can plot this Rle plot. So this is before batch correction, looks pretty good. After batch correction, looks also pretty good. So I'm not really sure that batch correction really did very much in this case. But this is how one way to perform simple batch correction if you have batch covariate information already. And there are alternative approaches. If you don't have this information, you can still correct for batch effects. And I will save that for another session. So the conclusion is Combat produces a slight improvement. You now see biological replicates clustering more tightly. I guess so. But anyway, so the scran normalization had the biggest impact, and it looks like Combat had a mild improvement on that. So in the final part of this talk, we will briefly discuss differential expression analysis. Now, differential expression analysis can be performed, as you can imagine, in various ways using single-cell RNA-seq data. For this session, we'll just consider one approach that is special to single-cell RNA-seq data. So from the book, they point this out. They say, unlike bulk RNA-seq, we generally have a large number of samples, so that is cells, for each group we are comparing in single-cell experiments. Thus, we can take advantage of the whole distribution of expression values in each group to identify differences between groups rather than only comparing estimates of mean expression, as is standard for bulk RNA-seq. Meaning, in this case, what we're going to do is we're not going to do our standard kind of t-test comparing 2 means. We are going to actually compare the distribution for each gene of the expression values between 2 samples. And we will see how we can do that. And the test that we'll be running to evaluate this difference between distributions is called the Kolmogorov-Smirnov test. There are multiple approaches to quantifying the similarity between 2 distributions, and this is a very popular approach. So before we go ahead and do that, conveniently, the book also supplies us with a list of true positives, so differences we know exist, and true negatives. Recall that this experiment contains 3 individuals. We'll be doing this differential expression analysis for 2 selected individuals. So if we go to line 204 and run that, this is a list, so actually a vector, DE. So these are all the genes that are differentially significantly different between these 2 individuals. So the 2 individuals we'll be looking at are NA19101 and NA19239. So DE are the genes that are known to be different between these two individuals. We also have a list of genes that are known to be not different. So that's the not DE. And if we look at line 206, we can assemble these two pieces of information into a list. And we call that list ground truth. And so this list just contains two elements, the DE and not DE. And if we go to line 211, we can extract the DE information from ground truth using the dollar symbol. So once again, these are all the genes that are known to be differentially expressed. And we can do the same for the genes that are known to not be differentially expressed. So this is useful information to have, to compare, to evaluate the results of our analysis. So this ground truth has been produced for the comparison of individual 19101 and 19239. So since our UMI.QC object contains information for 3 individuals, let's begin by discarding the information associated with the individual we don't need. So to do that, we can just type in UMI.QC, and once again, we use the square brackets notation. We want to keep all the rows, we want to keep all the genes, but we just want to retain information for these two named individuals. So the way to do that, one way is to do just UMI.QC $individual. So if we just were to run that in the console, we get for each cell information about which individual that cell belongs. So we can do this, and then we can use this percentage in percentage operator to ask the question, going cell by cell, does a given cell correspond to one of these two individuals? And to do that, we use this in operator and then NA19101 or NA19239 UMI. And then what we can do is we can assign this to a different single cell experiment object. So we can call that UMI underscore subset. So before I run that, let's take a quick look at UMI.QC $individual. Let's just remind ourselves of how many unique individuals our data set contains. So to do that, we type this in, and then we put that within this function called unique. So we can see that there are 3 unique individuals. Now let's run line 217. And then let's repeat what we did, the unique function, but let's now apply this to UMI underscore subset. We now see we just have 2 individuals. You might notice that if you look at the levels information, we still retain the levels information for the individual whose information we've discarded. This might seem a little bit confusing, but we would have to manually re-level this column to get rid of that level. And recall, re-levels or levels, these have to do with factors. I am pretty sure that even if we don't do this, the rest of the analysis we do will be fine. But if you want to be extra careful, you can you could just re-level this this variable so that it has 2 instead of 3 levels. But I'm going to skip that for now. Okay, so we've subset our object, and what we can do is, since we're applying, we will be performing this Kolmogorov-Smirnov test using our scran, our batch-corrected scran normalized data. We can extract that assay from our UMI subset object, and we can save it as a data frame. And to do that, it's easy enough. We go to line 219. To extract the combat assay, you just do assay, give the name of the single cell experiment object, and then the name of the assay. And then you have to convert that to a data frame. So actually previously I was using data frame in a few places where I really should have been using the term matrix. Not super important, but that's a correction. So if we run this, line 219, we now see that we have a data frame df. We run that in the console. We have, this is all information that corresponds to our batch corrected normalized data. And we'll use this we'll also extract the individual information, so on line 220, and save it as an object called group. So if we run group, we see that this is just a vector that contains the name of the individual. So in this case, there are just two individuals. So this gives you the name of the individual for the corresponding cell. We'll see why this is useful. Okay, so now we're ready to perform our Kolmogorov-Smirnov test. Now this is a little bit involved because we'll be discussing a few concepts that we haven't really gone into before. One is the concept of functions in R, and the second is a specific function called apply. Now, there's a general rule of thumb in programming that if you're going to do the same thing more than twice, you should write a function for it. Now, in this case, recall that we're looking for differences in gene expression for each gene between these two samples. And there are some 13,000 and something genes. So we definitely need to write a function. So let's look at this once again from the inside out. Let's start off with this on line 225. So to do a Kolmogorov-Smirnov test, we can use this function called ks.test. And I believe ks.test is just a base R function. It belongs to the stats package, which I think is part of base R because it's so frequently used. So the way the ks test works is it's really easy. What we want to do is we want to, so we begin, let's say, with we do begin with the first row, so the first gene. The ks test, we have to supply it with two things. One is the gene expressions for all the cells that correspond to individual 19101, and the second is the gene expression for all the cells that correspond to individual 19239. So the way we do that is, it's a little bit confusing here. There's this x thing because we're going to wrap this in a function. But we can initially just think of x as the first row of the df object, so meaning the first gene. And to extract just those cells that correspond to this individual, we can use this group information. So we can subset group equals this. So to kind of show you what I mean by that, so to extract the first row of df, we can run this. So this df square brackets 1 comma, so that's the first row, all the columns. Now, to just extract those cells that correspond to individual 19101, you can do group, you can further subset square using these square brackets and type in group equals NA19101. Now when we run this, it's kind of hard to tell, but all the samples here, notice that you only see 19101, right? Just 19101. And similarly, if you wanted to just get the cells that corresponded to the second individual, 19239, you can do the same thing, just change it to 19239. Now, if you look, all the cells correspond to this individual, the second individual. So that's it. And then we can just do this ks test using these two pieces of information. And the ks test will produce multiple outputs. So if I look at, yes, so if you look at the help section for ks test, you can see it produces a statistic, a p-value, a bunch of other things. We are just interested in the p-value for today. So we can just kind of chain it. So we do our Kolmogorov-Smirnov test, and then we extract the p-value. So that's what this function is doing. It is performing a Kolmogorov-Smirnov test and extracting a p-value. Unfortunately, we have to put this function, we have to do a little bit more work. This function, this ks test can only be applied, has to be applied row by row. So to do that, we take this function that we wrote here, so starting with function up to p-value, And then we put that, in turn, within another function called apply. And what apply does is it takes this function, applies it to an object, so our object is df, and to make it apply this function row by row, we specify 1 here. We could also, you can use the apply function and apply a function to columns, column by column, and then you would just do 2. You'll get a big error if you do 2 here. So if you do 1. So all of this, line 223, what it's doing is it's going row by row, computing the difference in distribution of gene expression for cells from this individual, 19101, and this individual, 19239, and extracting the p-value for each gene, and saving it as an object called p-vals. So if we run this, it takes a few seconds to run, hopefully not more than a few seconds. And now if we run p-vals, p-vals, we now have p-values for each of our genes, which is great. It's exactly what we want. Since we are performing multiple comparisons, we need to correct for this. And so if we go to line 234, that's easy to do. We can use this function called p.adjust, apply it to our uncorrected p-values, and we specify a method. Benjamin-Hochberg is a popular one. So we do method equals FDR, and then we overwrite p-vals. So now on line 235, we have an object called p-vals that contains our Benjamin-Hochberg corrected p-values for comparisons of distributions of each gene between these two individuals. So once again, from the book, this is to explain this line 223. This code applies the function to each row specified by 1 of the expression matrix data. Oh, this is a typo. So in the book, they call it data. I call it df. All right. In the function, we are returning just the p-value from the KS test output. We can now consider how many of the ground truth positive and negative DE genes are detected by the KS test. So this is exactly what we'll do in the last 3 to 4 minutes. So we can, on line 242, we can extract all of the significant genes. So meaning those genes that have a p-value less than 0.05. And we do that like this: names p-vals, and then we subset on p-vals less than 0.05. Now, I'm not going to go through this piece by piece. This is, I guess, a little bit of homework. Try to see if you can work out the logic of this. You should be able to, because this is similar to the kind of things we've done both today and previously. But if there's any confusion about this, please let me know. So if we run this and we type in sigDE, in the console, we get the names of all the genes that are significantly different between these two samples. And if you want to see how many, we just do length, sigDE. There are 4,752 genes. Okay. How many of these, so how many of the known differentially expressed genes, so our actual positives, are in this list? So we can do that on line 245. Recall that to get our known differences, we can use this list, ground truth, and then extract the differentially expressed genes, $DE. We ask how many of them are in this sigDE object, and we count the number. So we see that there are 781, and these are all the true positives. So these are known, these are genes that are known to be differentially expressed, that our Kolmogorov-Smirnov test says are actually differentially expressed, meaning it produced a p-value less than 0.05. Now let's see how many of the not differentially expressed genes are in this list. So it's line 246, we get 2,889. These are all false positives. So all of these genes have a p-value less than 0.05. But we know for a fact, because we've been given this information elsewhere, that these are not differentially expressed. So from the book, as you can see, many more of our ground truth negative genes were identified as differentially expressed by the KS test. So these are the false positives. Then the true positives. However, this may be due to the larger number of not differentially expressed genes. Thus, we typically normalize these counts as the true positive rate and false positive rate. So I really think it would be useful to dedicate a whole session to things like true positive rate, false positive rate, precision recall, confusion matrices, things like that, because these are all core concepts in machine learning. But just to quickly compute these various things, so true positive, true negative, false negative, we can compute the true positive rate as true positives divided by the true positives plus false negatives and the false positive rate. And if we look at these two things, we have a true positive rate of 75% and a false positive rate of 28%. So it's good. Our true positive rate is much higher than our false positive rate. I'm going to just rush through this last. We just have one chunk left, and I know I'm out of time. I am happy to go through the details of all of the code in this chunk offline, but I want to quickly run this. So the book makes the point that we are evaluating our Kolmogorov-Smirnov test based on a cutoff of 0.05, which is somewhat arbitrary. And in a lot of these like binary classification tasks, that's what you do. You specify some kind of cutoff. Anything below it belongs to class A, anything above it belongs to class B. What you can do is you can evaluate the performance of your model based on a continuum of cutoffs, which is what we are doing in these lines of code here, which I'm just glossing over. Point is, when you do that, when you compute these true positive rates and false positive rates for different, in this case, p-value thresholds, you can plot what's known as a receiver operating characteristic curve, which is this. And so the way to interpret this plot is if your model is just making guesses, so you will just get a diagonal line that's 45 degrees. If it's making perfect predictions, you will get a line that is kind of hugs the x-axis and this top of this sort of the roof of this plot. We're getting this curve here. And to kind of quantify the performance of our model, what is typically done is people quantify the area under this curve. So this is something that you may have seen elsewhere, this metric called AUROC. That stands for area under the receiver operating characteristic. If you get an AUROC of 0.5, your model is just making guesses. You might as well be flipping a coin. If you get an AUROC of 1, your model is making perfect predictions, so you should be suspicious. So if we run line 279, we can compute this ROC, which is called AUC here. And we can extract it, and we get 0.81, which is, as I say here, not bad. So that is it. We've covered a huge amount of material that really we could be dedicating multiple sessions to. But I hope you found this useful. Thank you for your attention, and I hope to see you in the next session. Thanks.